Thanks for hosting this today. First, I'd like to say how much we really appreciate this opportunity to be here and share our vision of the future. So this is a wonderful challenge that you give students to participate in growing that vision of NASA. This is the Serona team. We are the sustainable integration of regenerative outer space, nature, and agriculture. Serona is the Celtic goddess of healing waters, wellness, agriculture, and Serona translates into star. It was very fitting to use a female goddess that embodied these characteristics, as that is what our greenhouse is trying to represent. First, I'd like to introduce myself. I'm Heather Hobbit, and we're going to introduce the rest of the team.
My name is Sarah.
Kai. Abby.
Haley.
Lizzie. Larissa.
This is a wonderful team, and as you can see, we are all women, and it's a very unique feature to be able to have an opportunity to have a team of all women. We started out by designing a mission statement, and this drove the rest of the design of the greenhouse. Our mission statement is to design a greenhouse that improves habitability, human factors of the outpost beyond just satisfying the basic nutritional needs. If you see the normal acronym for BLISS, it's spelled without an I, but we integrate the I because a BLISS, as nature provides bliss, we can have life support and access to nature, and this is a very important aspect of our greenhouse design. I'd like to go over the design philosophy of how we approach this design. We integrated some of the best of many different philosophies. Some of my PhD thesis work is the Living System Center Design Framework, and you're going to see a snapshot of how this greenhouse embodies that framework. One of the aspects from permaculture that we used is something called a needs and yields analysis. If you can see here in this Venn diagram, we have the humans on one side and the plants on the other. Then each of these have different needs on each side of that. But they also provide a yield. In our design, there is no such thing as a waste. If there's an output, it must be an input to another system. And therefore, you will probably not hear us say waste too many times in this presentation, and that's a very important aspect. One example really quick here is that water is important to all living things. Water is needed by the humans, but it's also an output from the humans, and as I mentioned, not a waste. It's a yield from the humans, and that water can then be used to fulfill that need for the plants. And you're going to hear more about how we do that in a little bit. And as you can see, one other thing I'd like to point out is the care tasks. Humans have a need to have contact with nature, and plants obviously need care. The current visions that you see for greenhouses design have a purpose in getting to the next stage of development, and we're going to show you another vision. In these greenhouses, you can see that they're really optimized for plant growth and not so much for human interaction or for the humans to spend time in the greenhouse for recreational purposes. Now, if your only purpose is to provide nutrition and maximize that, this is a good solution. However, if you understand that some of the other benefits that plants provide go way beyond just the mass that they output in nutrition. Now we're going to get into the Serona Overview. Where's the pointer? As you can see, okay, well, if you start at the bottom of the clock, we have several different systems that we're going to go into detail. The heart of our system is an IMTA system, which I'll explain what that is momentarily. We also have some very unique features, including a bathroom and a kitchen, and this is very important because we want this to also function as a secondary habitat. We have a second floor, and this provides additional growth space, including a nursery and a lounge area. Now we'll talk about what is IMTA. IMTA is Integrated Multi-Trophic Aquaponics. You'll probably just hear us say IMTA or aquaponics going forward. It's a big, long acronym, and NASA loves acronyms, but it's also complicated. In an IMTA system, essentially what this is we use fish at different levels in the food chain. We also have shellfish and different aquatic crops, and we have 3 species of each. What this does is provide a much more stable ecosystem, including having filter feeders such as mussels and shellfish to eat the leftover fish food, so this increases the output of our system. In addition to that, this is paired with a nutrient recycling technology with ebb and flow and aquatic crops, which I'm going to get into the more details of that. But the pond here is one of the best features of the greenhouse, as it provides a relaxing setting for them to enjoy their meals. The grow beds that we are using is ebb and flow technology. This is typical to use with an aquaponics system and very robust. The way it works is that periodically the grow beds are flooded with water and then drained. The media that we're using is called grow stone, and this is a unique media. Grow stone is a recycled foamed glass. It's very lightweight, and it absorbs water and nutrients readily during a flood cycle and re-releases it to allow for very even moisture to the roots, but it also increases the aeration to the roots, which is very important for root health and to prevent disease states. We also have wicking beds. Wicking beds are sub-irrigated beds, and the reason that we use these in combination is because the wicking beds allow for capillary action to water the crops, and therefore if there are crops that don't like having their roots submerged, then this is another technology that we can use. It's also both of these are very robust with the grow stone. If there was a pump failure, these could be watered by the crew or the automation systems that you'll see shortly. In addition, we could transition both of these systems to a soil-based system using regolith with inedible biomass and biodigester affluent, which you'll hear more about shortly. One of the most unique technologies we have is called a Biowick. This is an adaptation of a system from Earth that comes out of permaculture known as the Watson wick. We've adapted it for spaceflight with the grow stone and some other unique features. A Biowick uses the gray and black water from the bathroom. It can also take in water from the kitchen as well to grow fruit trees and vining crops. This is a closed loop system. These are woody plants, and the microbes housed within the bed of the grow stone passively process the gray and black water. Therefore, there are no mechanical failure modes in this technology. In addition, there's lots of transpiration of water, and we see that we will recover that water through dehumidification and put it back into the IMTA system. I'd just like to point out that the bathroom does not actually have clear walls, but for the purposes of this, we wanted you to be able to see it. Here is a demonstration of the Biowick with a prototype. In the video, you can see the infiltrator, as well as you can see the infiltrator on the model. The infiltrator is a large volume for surge capacity for things like showering or doing laundry to accommodate that water as it infiltrates out into the media. The reason we chose to build this as a prototype is that all of the grow beds can be designed with a similar type of deployable mechanism. This particular prototype is made out of food-safe materials and will be used to do some initial testing in a growth chamber when we get back from this project. And now I'd like to pass this on to our next presenter, Haley.
These are our Photobio Bioreactors. They utilize full spectrum horticulture LEDs, and they aid in the transpiration and respiration processes, as well as provide food for the 4 crew members. They provide about 10 grams of, they also grow our microalgae, which is spirulina, which provides about 10 grams of nutrients per crew member per day, as well as absorb 3% of the carbon dioxide per day per crew member, and then release about 2% of the oxygen per crew member per day. This is our prototype. Beware your eyes, it might be a little bright. The idea is that in deployment, these will just come up like so. And stick up there.
So one of the other unique parts of the Sirona design is that we have included two different types of lighting systems. The first lighting system, the LED system, is, as we've heard about from all of the groups previously, fairly power intensive. It requires a lot of the energy allocated. Our secondary system, which I'll talk about in a second, is a Himawari solar collector system, which is actually a much lower power requirement system that makes use of natural lighting. But for the LED system, which you can see here, it's from Acro Optics, and it's a fully adjustable, programmable LED system that has both UV and IR radiation, which is important for crops at different times in their life, for fruiting versus vegetative growth, for example. It's important that they're adjustable, because one of the things we intend to do is manipulate the environment, the light environment in which our crops are growing, to increase the nutritional quality, specifically the carotenoids, which are photoprotective pigments produced by a lot of plants, and are also good for humans in terms of eye health. The Himawari system, as I said, is a lower, it requires less energy, and it basically has, you have an array of solar fiber optic lighting connected to collection cells that then funnel down natural lighting from the Martian world into our system for a more passive light supplement. Also, one of our, so for our environmental controls, one of the organizational design factors in the Serona system is that we've split it into two major terrestrial zones, Zone 1 and Zone 2, which are, for our terrestrial plants, mean that we have some nod of the hat, if you will, to adaptation that's occurred on this planet. So we have crops growing in Zone 1 that are more temperate, came from more temperate environments on Earth, and crops growing in Zone 2 that are perhaps a few different, that can grow in a broader number of environments. But we've basically matched day temperature, night temperature, light duration to some extent, and our vapor pressure deficit and relative humidity to those Earth analog ecosystems.
So what do you do with all of that yield that comes from our growth systems? Altogether, whether it's our fresh crops or the resupplies and the packaged foods, probably 7 million calories is going to be consumed by our 4 crew members over the course of the 600 days. That's a lot of food, whether it's just heating it up or cooking it. We don't want our crew to be only eating raw salad all the time. So we want our food to be nutritious and palatable. And for all of those reasons, we decided to move the small kitchen originally attributed to the ice home into Serona, into the greenhouse, because, I mean, we all enjoy cookouts. We all enjoy sitting outside, being in nature. There was the mention of the concept of biophilia. And to do that, we took a tip from the world of professional kitchens and restaurants. I would argue probably that restaurants in New York, that some restaurants probably are more conscientious about mass power volume than NASA even. And so to that end, we took a trick from them, which is how they design kitchens, is that for every station, for each chef, the station is set up such that they barely have to move. They don't actually have to step out, and they can just reach high, low to get what they need, because we need to be conscientious about space in the greenhouse. To that end, we have most of our processing, rough processing, lining the wall, the periphery of the greenhouse. And then the center kitchen island has lowboy freezers and refrigerators, and we have equipment such as sous vide baths for low temperature cooking, and ovens that can bake as well as dehydrate. So it's versatile for a large variety of post-harvest processing. With that said, we wanted to be functional, certainly, but we also wanted to be a place where the crew members could just hang out and bond with each other. And that is good for social cohesion as well as mental well-being. I'll point out one specific technology in the kitchen, which is the biodigester, which is connected to the sink. For inedible biomass, a lot of perhaps the tops of For a lot of an edible biomass, that can be macerated and goes down the sink and then goes into the biodigester, which basically works like a cow's stomach. There's anaerobic bacteria in there that process the organic waste to produce a fertilizer. And it also produces biogas, half of which is roughly methane, the other half being carbon dioxide, and that biogas can run the oven. And in a pinch, the methane can also be hooked up to a generator to provide backup power.
So one thing that both Larissa and Heather and I to some extent have already spoken about is the resiliency that we've built into our system, the robustness of including humans in the design. So another way that we included humans in the Serona crop design was through the selection process. As you can see down at the bottom of the screen, we have a lot of crops that we intend to grow in this system. And by increasing the diversity of yields, we intend to increase the resiliency of outputs overall. Broadly, our terrestrial crops fall into a few different categories. We have leafy greens, roots, fruits, herbs, and then we have 4 aquatic crops and 6 freshwater seafood species to supplement nutrition and also functionality in the system. Another part of this system that puts the astronaut, the crew member, into the design is that we have a plan for a more long-term crop breeding program that includes the astronaut in an adaptive breeding program. Here on Earth, it's called participatory plant breeding. In a Martian setting, it's the only plant breeding program I can think of that would work. So to get to that shortened list that I just presented to you, this is the complete table. You might be looking at that like, wow, that's a lot of crops. That's a lot of things that could go wrong. But in fact, that's possible, and you'll hear about the risk mitigation strategies that we took. But actually, by including so many different crops, I think that we actually have an eye towards overall robustness in the system. We arrived at this table through a sort of a new yield metric called Composite Yield, which I'd be happy to talk more about in the question and answer period. But essentially, it values each crop for more than just its biomass production. It's also, in this system, nutritional, psychological, and overall dietary value. But before we actually start growing these things and reap those benefits, we have to get our crops to Mars, which is no small feat. Phase 0 for crop deployment will be, of course, transit, and we intend to transit in the dormant phase, both in the vertebrate and plant crops. Once the crew arrives, from 0 to 6 months, that will be phase 1, in which the yield will be microalgae mostly and microgreens, anything short with a short life cycle. Phase 2 is from 6 to 12 months. Those are, in addition to the microalgae and microgreens, we will also have some fish. We'll have potentially the dwarf plum that Ray actually talked about earlier, could start maturing in that first year, definitely in phase 3, as well as some of the other smaller annuals. And then finally, phase 3, in which our food forest growing in the Biowick, those woody perennial fruits, start to mature, and we have our full palette of crops for the astronauts to eat.
So you might be asking yourself, are they there to be an astronaut or a farmer? Or are they astronaut farmers or maybe robot farmers, or do we need both? And that's what I want to talk about next. Of course, in a system like this, as you saw with the many crops, there's lots and lots of care tasks to do, and astronaut time is the most premium resource on a mission, much even more so than a power. Power you can get more of through a lot of engineering work, but you only have a finite number of hours in the day, which is the great equalizer in life. Roger is the remotely operated gardening rover, and Roger was originally developed by the CU in the XHAB competition of 2014, including SPOT and AgQ. And so we've been developing these systems for some time now. You can also see how Roger can tend to the beds in the render on the other side. Roger can do many tasks, but some of the things we envision on a routine basis would be seed germination, transplanting and pollination, and dealing with inedible biomass. However, if the crew gets too busy, Roger will be able to take on other care tasks. In addition, Roger will do all of the imaging necessary to monitor plant health. In addition to Roger, I mentioned AgQ. AgQ is the plant care decision support software. And essentially AgQ works by taking in data from several different sensors that monitor plant health. I'm not going to go into the litany of sensors, but believe me, we have plenty of them. In addition, we're also monitoring human health, their physiological stress state. What we want to do is have the crew be able to take a break when they're stressed out, if they're doing a non-critical task like writing a report, for instance, and go and do some horticulture task that might provide the benefit of interacting with nature to have horticulture therapy. As I mentioned, Roger will be also doing lots of imaging, including infrared, stereoscopic, and visual spectrum. All of this data is fed into the data pipeline that uses machine learning and a lot of other tools. It does monitoring, alerting, control, and prediction. Eventually we'll be able to do trends and prediction to catch issues before they actually occur or early on. With this type of visual imaging system, we can see crop disease or nutrient deficiencies before the human eye can detect them.
Okay, so there's two types of tasks that the crew would be doing. In the greenhouse, there are the daily tasks and the infrequent tasks. The daily tasks include crop care, such as thinning, pruning, harvesting, quality checkout, and cleaning. And the infrequent tasks include things like sanitizing equipment or calibrating sensors. These infrequent tasks needs to be done every like 90 or 180 days, depending on the tasks. The crew would be spending on average 20 minutes a day per crew. And Roger would save about 140 minutes a day in equivalent crew time. This is the deployment timeline. It has checkpoints. So phase 1 lists the pre-crew arrival, and it majorly consists of the structure deployment and the floor deployment. And phase 2 is after crew arrival, and it consists of making sure that everything deployed correctly, deploying the grow beds, filling the water, or installing lights.
So one of the big challenges for us was to figure out what the deployment of the second floor was going to be, especially because we are suggesting it supports a lot of elements in the design. After a lot of deliberation and consulting, we concluded that the best way to go for us is an inflatable second floor, and we are intending to demonstrate the deployment of that second floor. We try to mimic the blooming of a flower with our petals and then inflating them. Here we take a more modular approach where we first latch all of the petals and then inflate them. But here I'm going to demonstrate how we deploy it as if it were a flower.
So I want to talk to you guys a little bit about risks in the Serona system. So we selected subsystems that were low rates of risk and failure. Also, we designed the Serona system to act as a secondary habitat in case of any emergencies that happened in the Mars ice home. As Heather mentioned earlier, there's a bathroom and the walls aren't transparent, they're there. And we also have a kitchen and emergency sleeping quarters that are usually stowed away during normal greenhouse operations. And also we have a slide. It's an emergency slide on the second floor in case the stairs are inaccessible. So who doesn't want to slide in their greenhouse? We also did analysis of 20 different risks, and an example of one that we thought was important was loss of fish in our system. And in that case, we have hydroponic nutrients and also biodigester effluent that we can substitute as fertilizer. Here you can see our power and mass tables, or pie charts that we created after a lot of analysis. I like to point out here, if we used 100% of the power, we would have an 11.6% power margin. If we used 100% of the power with lighting and then 50% for other equipment, which is more realistic because we're not going to be using 100% of the power, we would actually have a 30% power margin. And for mass, we have a 16.2% margin. Also, as Lizzie pointed out earlier, the Himawari and LED systems sort of balance each other out. The Himawari has a lot of mass, but uses less power, while LEDs use more power, but less mass. So here you can see our entire system overview. So we went through all of the technologies, and here is how they all come together and how they interact with each other. As you can see, it's a closed loop system. So everything in green is our land crops, and they have a total surface area of 51 meters squared. In blue, we have our IMTA aquaponics system, and it has a total volume of 7.32 meters cubed. Everything in the dotted lines, the green dotted lines, is our food production output for the crew. In black, we have our overflow and black and gray water systems. You can also see in the gray are the list of sensors that Heather was talking about earlier for AgQ.
So we've just seen how Sirona's subsystems interact with each other. Now we'll take a look at a summary of Sirona's inputs and outputs. This summary effectively demonstrates the efficiency of the overall Sirona system. So Sirona can meet many human requirements. Sirona provides approximately 98% of the O2 production required, approximately 74% of the CO2 uptake requirement, and can provide 100% of the drinking water necessary for a crew of 4. The Biowick needs 140 liters of water per day to sustain the crops that grow within it. So the crew will provide an estimated 40 liters of gray and black water per day. This means that the Biowick is capable of essentially processing 355% of the estimated bathroom wastewater. So the remainder of that Biowick water and nutrient requirement can be provided by the inedible biomass and water refuse from the kitchen or by the aquaponics system. So the total daily water uptake by crops in our system is estimated to be approximately 258 liters. It is expected that 94 to 99% of the water transpired is recovered by dehumidification into the aquaponics system.
And finally, let's look at Serona in terms of the diet. We're averaging about 30 kilograms of edible biomass per square meter. What that translates into is about 1.3 kilograms of food. That's fresh fruits and vegetables and fish and seafood for our crew members every day. That's about 3 pounds. That's about 20% of the target, 3,000 calories for each crew member. And that's as expected because we know it's realistic that Serona or this greenhouse is not going to provide all of the entire caloric energy required during the mission due to the current state of LED technology. We're meeting about 30% of the protein target and of the carbohydrate target. And of the key 24 nutrients identified by the USDA, we're able to provide an excess of 50% of the daily target amount for 11 of those nutrients. So with all of that said, 2 years on Mars, would you like to stay here or here? And we'll give you a better sense of what it would be like to spend time in Serona with this fly-through. It's a little fast, so apologies if it makes you a little motion sick, but it'll finish soon. And we use this presentation to make some of the things that don't jump out from the paper, from the PDF, to highlight those aspects. But we have our tables, our extensive calculations on hand and backup slides, so we're happy to delve into that. And finally, we want to give a huge thanks to a number of people, especially to the Big Idea Challenge for providing this platform, to CU Boulder, to Harvard, to Big Idea for funding, to our respective institutions for and our professors, our advisors for their support, and a number of family, friends, colleagues for helping us to get here. And now we're happy to answer your questions.
Hello? Okay. Thank you so much for your presentation. Really wonderfully comprehensive and very interesting human factors, especially approach. And I'm wondering, sort of as a very zoomed out question, is you use a lot of passive systems, complex systems, and you're trying to make an entirely closed loop mostly bioregenerative system. And I wonder if you would describe or talk about the challenges in sending and convincing people to send this rather than a what others might consider low risk system or a very conservative system. So yeah. I think that will be a major challenge for you, so I wonder how you address that question.
So actually this is low risk in a lot of ways. As I mentioned, passive systems do not have mechanical failures. So if you're concerned about launch mass, now you don't have to send triplicates of every spare part. However, parts that we have, of course we do have pumps and things like that. We anticipate that those are designed to be 3D printed as well as sending them some spares. But the more that you can reduce mechanical systems, if you already have living systems anyway that we would all have the same kinds of risks in some aspect of that, you can reduce the overall risk by reducing the number of mechanical systems. One other example is by having two different types of lighting systems. When the power system might go down, we can still provide some lighting through the Himmelry with backup power by just using the solar tracking of that system. And of course, we still have the optimization of being able to control the light source. So those are a few ways that we've reduced risk by the particular technologies we selected. And I'd just like to finally mention that those grow beds, if we did have complete pump failure, because of the grow stone, we can have Roger or the crew hand water them because that actually doesn't need to be watered as frequently. So if you have something like NFT, your mean time between the crops dying is about 15 minutes with a pump failure. And so to me, that's a much more risky system than having hours in between needing a watering. Yep.
Just really briefly to address also your question about convincing people to send our system. I think that's a really good question, and I think, so I'm a plant biologist, I'm an ecologist, so part of what I find compelling about this is that it's a microcosm of what we experience here on Earth, only we get to redefine the rules, if you will. So in that way, it's an incredibly attractive system to send to Mars because in the long term, what we actually want is for this system to be one of many in the evolution of Mars plant growth.
Thank you very much. It's pretty noble to look at a system where there's no waste, and effectively you're seeking 100% recycling, right? That's what we want in a fully regenerative, a fully autonomous, a fully sustainable spacecraft. I mean, that's where we want to be, right? What's interesting is that although you're essentially fully closed with respect to waste recycling, you're not really fully closed with respect to the humans and meeting their needs, because I think you're only at about 20% of the calories, right? I think, right? And but I think that's kind of where we want to go, especially on Mars, where you have planetary protection is an issue. It's like, leave nothing behind, right? You're making use of just about everything. To define something as there's no wastes here, right? Even though we might say it's waste, it's really an effluent or an input to something else, which I think is a pretty neat concept. When I look at this, I think of it as a very simple system from a mechanical and an engineering point of view, but I see it as a complex from a biological point of view. Would you agree with that?
Yes, and I'd just like to say, We didn't necessarily set out to close the loop on everything. We started with crop selection and the, as I mentioned, living system center design process. And then after we got into the detailed analysis, we saw the performance of the system was approaching that. Of course, we looked for technologies that would help us close the loop to some certain extent. But we think it's very realistic to send high-calorie staple crops, because I think that when you're dealing with things like wheat or other things that have lots of inedible biomass that have long shelf lives, there's no real advantage. The real advantage to crops that we're growing is the fresh palatability and the variety, and that was the focus, not necessarily calories. And we set out with that focus not to provide calories, because calories are easy to provide through stored food. As far as being, I think the second part of your question was the mechanical system being simple, but and that was part of what we wanted to do. I think all biological systems have failure modes, but we anticipate that we will test all of these things on the Deep Space Gateway first to ensure that they're going to survive the transit and long-term testing. And all of these systems would have to be tested that way in any case. And so I think that if you're going to send a biological system, you're going to just accept that type of risk. And you're going to have physiochemical backup systems in the ice home. One unique feature for us that we've reduced the risk, because no other team here has provided a full secondary habitat with all the functions that the ice home also provides. And so that reduces the overall mission risk in totality.
I'll just add one more thing about the caloric energy that we're providing. And you might look at the crop complement list that Lizzie showed, and it was a very big list, and you wonder, why do we have nasturtiums on there, for example? And the point, to back up what Heather said, is that staple crops we can ship. It's when you're on day 300 and you're missing the crunch of an apple, you're missing the crispiness of carrots, that this is where Serona comes in. I mean, it's not just that, it's providing the caloric, it's providing 20% of the caloric energy, it's providing a lot of the micronutrients, because a lot of micronutrients degrade during transit too. So you have continuous access to that. And then in terms of this, my tangent about the nasturtiums, it's because we used, and Lizzie can explain this, we used this composite yield metric to quantify certain crops that might not make it on there if we're only looking at their caloric value, but brings medicinal value or psychological benefit.
Okay, good.
Sorry, I just want one quick follow-up. Is it a scale issue? If this were bigger, could it get more calories, or is
Yes.
it an issue about also
I think
staple crops?
Yeah, I think definitely in the future, developing the system to grow both other crops and grow more of the crops that we've suggested here, that would just change a lot of the metrics in the composite yield calculation, actually. One thing about the composite yield calculation is that it's probably adjustable. It's definitely adjustable, and that's something we'll actually talk about if you'd like. Yeah, there you go. So there's our composite yield calculation in its simplest form. All of those factors are adjustable based on what your priorities are for the system.
Also, really quick, I just want to say that these technologies can grow. We selected these crops because we looked at what a lot of the dietary requirements were and what has been set out by NASA guidelines in terms of meal planning, and we used that to determine how much of each crop we should grow if we were selecting that crop. However, these technologies are very diverse and can grow many, many other crops, so you don't have to use this crop complement. It's just our suggestion to do the analysis, so you could grow whatever crops you want with these technologies because they're that diverse.
So I want to commend you with the diversity aspect in particular. It seems like an extremely complex system precisely for the diversity of agrosystems that you've introduced. My first question is whether you've considered, or in what way you've considered, the relationship of these systems and why each has been sized and scaled in the way that they have and how they work together as opposed to separately. And then my second question has to do with the deployment of the interior systems and materials and structure overall, and whether you have any sort of, whether you've given any thought to that. I noticed that you had a high-level timeline for the deployment of elements, but I mean, when we're talking about materials and fixtures and plumbing and mechanical, I mean, I'm just curious how this would all come together.
Sorry, could you just briefly repeat the first part of your question,
Yeah,
Sorry,
let's start with the
'cause you
first
asked
question.
two different questions that I...
The first question had to do with the diversity of
Okay,
the
got it.
agrisystem.
Okay, so one thing about the diversity of the crops. So I just want to say, does anybody want to eat 8 crops for 300 or 600 days? There's something called menu fatigue, and right now astronauts already experience this with the diverse foods that they have provided at this point in time, and they experience space anorexia. So can you imagine eating 8 crops for 600 days? Do you ever eat 8 foods for your entire diet? It's just not realistic in terms of dietary perspective, and Larissa could talk more about that, but I will answer your second question if you want to follow up from her. The second question was about the plumbing and the deployment aspects. Was that correct?
Well, I'm less interested in the crop profiles that you've selected and more interested in why you've introduced aquaponics with the other systems, for example, or in what combination, or whether you've considered whether one bioregenerative system would be sufficient to yield the kind of crops that you want.
Sure. As we mentioned, if the fish were to die, we've already provisioned on the mass profile that we're sending all the necessarily hydroponic nutrients to feed the crops in addition to the effluent, and therefore these tanks, if there's not fish swimming in them, one of the principles of permaculture is to stack functions, meaning every element provides multiple functions, and so in that case you have water tanks that you have to have anyway, you might as well, and if they're going to have nutrients, why not that nutrients come from fish? And also the other part of it is every function should have multiple elements that are achieve that, and that is how we have designed this system. So you can see that with these different functions within the different elements providing that capability. Does anybody want to follow up on that?
I also just wanted to answer your second question as well. So with deployment, most of our structures, as you could see, like with the grow beds, they're deployable. This can also be taken apart, and this can come down. But anything that cannot be collapsed will go in this center column here, where there's space, because these will fold up, and that's how we'll have the things that won't be compressed.
I thought this was really unique in terms of your approach and philosophy of considering habitability and human factors. So that I thought that's something very commendable, as was mentioned. When you kind of integrate, say, the kitchen functions and maybe prolonged habitation in a plant growth environment, how would you deal with humidity, for example? Plants like it humid, humans don't like it humid. That was one aspect, and also then just trace contaminant control in terms of your integrated functions. You have to deal with it in habitats anyway, but you're also then taking this on in this system as well.
So in regards to the humidity question first, you are correct. Humans don't like 80% humidity all of the time, neither do mechanical components necessarily. So our waterfall, for one thing, we have a lot of plants, which, as we've learned about, there's a lot of water cycling in general with the plants, but we also have this waterfall, which will be part of the dehumidification, and we have a dehumidification array also that was mentioned earlier. In terms of keeping humidity high enough that crops find it acceptable, that's part of our zoning scheme. So those crops that are adapted to especially high humidity levels on Earth will be in the higher humidity level zone, whereas those that are more tolerant of mesic conditions will be in our lower humidity zone.
And then also, go ahead, but the other volatile organics, if you have your bioreactor, biodigester, your Biowick system, I mean, it's generating syngas. How do you manage those kinds of things?
I think that's a really fascinating question. We have a few different volatiles coming off of organic matter generally, right, including the biodigester, which potentially could ripen some of our crops or change the succession of our crops in general. This is something that definitely could be included in the design in terms of circulating the air, but I think it's actually probably a really interesting place to start doing some future research to include other volatile organisms or volatile compounds that are off-gassed by plants on Earth, because they'll all be collecting in this environment. And even though they might not be perceptible to the crew, the plants might be picking up on signaling through that. So that would definitely be a fascinating thing to research.
Just quickly, we will of course have air scrubbing to take out those volatiles. We didn't go into big depth about that because there's just not time in this presentation. I think the other questions should be answered first.
Right, about your question of trace contaminants and cooking in this area. Can we go to the slide with an overview of Serona? Maybe the last. Sure. Yeah, right. So we put the kitchen where it is for a reason, and we swapped out the kitchen that is in the ice home. I think maybe we did not discuss this in the paper, but this is our idea, that in the ice home there is a small commissary of sorts, small kitchen. The full kitchen is here. Why? Because it's easier. You're harvesting things in the greenhouse, then your things are perhaps, I don't know, dripping in dirt or something, and it's easier to just bring it straight away to the post-processing area. Now we put it there, so there is, and then there's the recreational lounge area in the middle. So there's a little bit of a separation between where you're cooking and eating and the rest of the growth space. We put most of the rough processing, that initial cleaning, washing. Okay, right there. So that is the rough processing sink, and that is connected to the biodigester. So right away, where the kitchen, a small part of the kitchen will get dirty, literally meaning it gets dirt, but we don't expect that and to influence the rest of the kitchen. And so the crew, in the middle of the night, they want a snack because they're living in the ice home. Therefore, they have the kitchen, that small kitchen in the ice home. They do not have to go all the way into the full kitchen to get at the food or to cook the food.
Since you're the first ones to have seafood here, how long will it take before the first sort of seafood meal is available to the crew, and at what rate is that produced? I'm assuming that you have to, it's not fair if only one person gets a bite, so you have to kind of batch it as well, right?
Yeah, good question. So the 3 species of swimming fish, finned fish, barramundi, jade perch, and tilapia all mature within about 6 months of once you've basically allow them to start growing, and you can keep them you kind of stunt their growth during transit because you don't want them to mature. We're not planning on bringing the entire amount that they would need. We've only selected things that will breed in captivity, including the crayfish and the giant prawn. These are also species that can cohabitate in an IMTA system. We reviewed a lot of the literature of what species do cohabitate. So in Larissa, I know that we, the exact amount of fish, it was like, it's 2 to 4 seafood meals per week of 8 ounces once you get to full maturity. The shellfish will be matured at the 12-month mark and start to be harvested, and it comes out to be about 200 kilograms per species, with the exception of a couple species, but that's a rough, and I'm not sure whether we can pull up the details if you're interested, but that's essentially what it is.
I'm gonna hope I'm asking Kevin's question. On the flower petal deploying, I think I understood, were you telling us that it didn't go so well initially, and so because of the way the prototype behaved, you decided to clip them together before inflating in the future?
So we started considering a rigid structure, and that is why it took us a couple of iterations to figure out the inflatable structure design. And the reason for that is because so when we estimated the mass that will take us to bring those structures, it was way above what we wanted it to be, And also because we wanted to seize the inflation stages of the greenhouse. Another reason that we picked inflatable structures over rigid ones is because we came across a couple of studies made by, one was a PhD thesis, two were from NASA, and they developed a couple of techniques that such as rigidization after the structure is inflated, that we consider were feasible for what we wanted. And also because inflatable structures are as lightweight as the greenhouse, so we could introduce all of these subsystems apart from the major structural components.
We have 17 seconds. One quick question.
A great vision for deep space exploration. Looks like you pushed the envelope on the flexible workspace and had everything, including the kitchen sink. So, but anyway, on the fish, you said you had it would require 3 cubic meters or 3 metric tons of water to transport them to Mars. I guess that'll come on a follow-up with the crew on a follow-up mission. You also had fruit trees that come. Did you do a mass estimate on all these additional things that need to be brought?
Yes, we did, and the trees are dwarf. I just want to emphasize dwarf as in smaller than me and transported in a dormant phase, so they don't require a lot of things to take care of them, and I know we're out of time, so I'll stop there.
Yeah, did you talk to any fish farmers or anything like that? Okay.
I did. I'd be happy to talk to you about that later.
All right, good job, guys. Serona. And we're going to take a quick 15-minute break, so please be back in your seats at 3:10 for our final presentation.